The process of cloning a gene involves insertion of a DNA sequence into a piece of autonomously replicating DNA, such as a plasmid. The transfer of this new plasmid construct into a bacterial cell, and the growth of a population of bacterial cells containing the plasmid. In this way, a clone is generated consisting of many exact copies of the inserted sequence. A plasmid contains a bacterial origin of replication that is required for it to replicate in a bacterial cell. The plasmid also carries a selectable marker gene that makes the bacterial cell transformed with this plasmid, able to grow in a specific defined medium. In our example here, the plasmid contains a gene that makes the bacteria resistant to the antibiotic ampicillin. In another location in the plasmid is a copy of the LAC-Z gene that will produce the enzyme beta-galactosidase when the plasmid is inside the bacterial cell. The LAC-Z gene in the plasmid has a region that is recognized by specific restriction endonucleases. Each restriction endonuclease that recognizes this region cuts the plasmid only in this location, so it is a unique site for that enzyme. Most restriction enzymes cut the two DNA strands in different places and thus produce regions of unpaired sequence called overhangs or sticky ends. The piece of DNA we wish to clone must now be generated. In our example here, we will clone the beta-globin gene obtained from human chromosomal DNA. If we treat human chromosomal DNA with the same restriction endonuclease used to open the cloning vector plasmid, a set of specific fragments will be generated that vary in size. Provided that there is not a restriction site inside the beta-globin gene, one of the fragments generated will contain the complete beta-globin gene. However, other fragments of human DNA not containing the beta-globin gene will also be generated. When the fragments of human chromosomal DNA are mixed with the plasmids that have been linearized by the restriction endonuclease, the sticky ends of the plasmid DNA and the human DNA fragments can hybridize with each other, forming double helical structures at the overhangs. These structures can be further stabilized using the enzyme DNA ligase. This enzyme ligates DNA strands by covalently connecting breaks that occur in the DNA backbone. This is exactly the situation at the sites where the double-strand overhangs have now hybridized. DNA ligase closes these strand breaks, making a new circular DNA cloning vector molecule with a human DNA fragment ligated into its unique restriction site. While some plasmids will receive the human DNA fragment containing the beta-globin gene, other plasmids will be generated containing other fragments of human DNA that do not contain the beta-globin gene. Still, other plasmids will simply have sticky ends that find each other and no human DNA will be inserted. DNA ligase will then close these plasmids again, regenerating the original cloning vector. Thus, at the end of the ligation reaction, 3 types of cloning vector plasmids will be formed. The population of various plasmids is then transformed into a population of bacterial cells that have been specially treated to make them competent to receive DNA. This generates a population of transformed bacterial cells which contain cloning vector plasmids with or without human DNA inserts. These cells are then plated onto solid growth media containing ampicillin. Only cells that have taken up a plasmid are able to grow and form bacterial colonies. Petri dishes filled with solid growth media plus ampicillin also contain a colorless substance that the LAC-Z gene protein, namely beta-galactosidase, can convert to a blue substance. After overnight growth, those colonies that arise from cells that received a plasmid without an insert will appear blue. This is because the LAC-Z gene in these plasmids is able to produce a functional beta-galactosidase. In contrast, colonies that received a plasmid with a human DNA insert will be unable to synthesize beta-galactosidase because the human DNA fragment would disrupt the connection between the LAC-Z gene and its promoter. These colonies will appear white. White colonies can then be selected and examined for the presence of human beta-globin gene. A colony containing this gene can then be saved and grown for further study of the cloned beta-globin gene.